and actually we are continuing right away with the keynote lecture of Associate Professor Volker Laushke, who is a group leader in personalized medicine and drug development and director of the Micro and Nanoengineering Faculty of Karolinska Institute. Professor Laushke, the floor is yours.
Thank you very much. So I seem to have some issues here with sharing my slides. The button is gray, but since
just
I sent
a second,
now it works.
will,
Perfect, yeah. Excellent, very good. So let me see that. Share. Can you see my slides?
Yes, now it's full presenting mode.
Brilliant. Okay, perfect. Yeah, thank you very much for your invitation to present our work and the work of others with some context about human genetics and personalized medicine. And I think, as you rightly say, this is really an amazing and exciting opportunity of collaboration and further interaction that we are having here. And if there's anything I can contribute to, then that would be my absolute pleasure. So let me start by highlighting the importance of genetic factors. So today I will primarily speak about pharmacogenomics. As the majority of the audience might know, there is a large heterogeneity in the response of patients to pharmacological interventions, up to the extent that estimates assess that between 25 and 50% of individuals actually don't respond as intended to treatment, which can issue itself either as a lack of response or adverse reactions. And the bulk of these causes are indeed due to non-genetic factors, drug-drug interaction, various physiological and pathophysiological factors, environmental factors, smoking, etc., as well as drug adherence is a major issue. However, there is around 20 to 30% of this variability that is indeed caused by genetic factors, and as such might be both predictable and preventable. And those genetic factors can then be further subgrouped into a variety of different variations in ADME genes, ADME standing for absorption, distribution, metabolism, and excretion of drugs. So that enzymes that are involved in drug metabolism or drug transporters might have altered activity and therefore either metabolize the compound too much or too little, resulting in too high or too low exposure to the compound. There's variability and variation in drug targets, which might then increase or reduce the efficacy and the pharmacodynamic interactions. And there's also an important additional category, which is the variation in genes that encode the major histocompatibility complex. And this is primarily HLA genes, and those cause, or can at least cause, altered immune responses and immunological adverse events. And I will go a little bit into detail there further as well. So again, this is an introductory overview of those genetic variants that can alter pharmacokinetics. So, for instance, if the compound that is administered is actually active as a parent, then increased functionality of an enzyme might result in decreased exposures. You have faster biotransformation and therefore faster inactivation of those active parent compounds. Inversely, if the actual metabolite is active, then you have an increased functionality enzyme will result in increased exposure, and vice versa for reduced or loss of function variants. So reduced function variants for an active parent will increase exposure and inversely decrease exposure for if the metabolite is active. Altered pharmacodynamics here, and also there we'll provide some more details later on, just so that we are all on the same page of what I actually mean with these, is that this is particularly used in oncology, that only those variants that have a mutated binding sites are actually targeted by a given compound. So this is used in targeted oncology quite extensively. And I will present also some evidence for the former here, that once there's a mutation in an actual drug binding site, that this will reduce the activity of the molecule that is intended to have on-target interaction here. And lastly, for altered immune responses, so there are a variety of different HLA alleles that slightly can change the shape of the major histocompatibility complex on the antigen-presenting cells. and therefore avoid the self-recognition of the T cells, which result in major autoimmunity-like interactions, specifically issued as Stevens-Johnson syndrome and toxic epidermal necrolysis. These are the major causes of morbidity and mortality in HLA-related pharmacogenomics, but also Abacavir hypersensitivity syndrome. So, as you likely know, over the last few decades or so, there have been a large multitude of associations that have been presented. This is just a short selection here. Various genetic variants in cytochrome P450s, DPYD is or altered DPYD activity variants have been majorly in the spotlight in recent years due to important work done by Henke and Guchela and colleagues in Leiden, as one example, showing really the added value of preemptive pharmacogenetic testing of DPYD to predict fluoropyrimidine toxicities. Similarly, CYP2D6 duplication and deficiencies, which are very common in most populations, can have major impacts on codeine efficacy and response. CYP2B6, another example. Various phase 2 enzymes as well, drug transporters and HLA genes. This is all toxicity associations, and similarly, there are a multitude of associations reported on the efficacy branch. So, for instance, warfarin metabolism affecting the response, and CYP2C9 and VCOC1 variants affecting the response to warfarin. Clopidogrel, another one. So I will not go into detail of these established associations and interactions and biomarkers. They have been reviewed extensively and are available for those that are interested. And of course, I would be happy to discuss afterwards in case there are any questions. But I would rather like to focus on more recent research that came up in the last 5 years or so. And a major breakthrough in this regard was the availability of large-scale populations as genome sequencing projects. So this is showing data from more than 60,000 individuals aggregated by the Exome Aggregation Consortium, covering a total of 208 different genes. And it really shows the extensive variability and number of variants that are present in pharmacogenes. So here we analyze specifically SLC, ABC transporters, CYPs, and other phase 1 enzymes, various phase 2 enzymes, and also some nuclear receptors. And at the time of the writing of this paper, really the vast number of those variants were actually novel. So overall, we identified around 70,000 variants, of which more than 50,000 were novel and not functionally characterized. And even among the known variants here, the actual evidence of what those variants are meaning for the drug metabolizing phenotype of the carrying individual are not clear. So there's really only a small subset of those variants, a tiny subset of those variants that is functionally understood. When it comes to pharmacokinetic genes, there are two further important things that I would like to highlight. On one side, the very low evolutionary pressure and evolutionary conservation. So here higher scores means more evolutionary pressure and more selection. And as you can see, the evolutionary pressure and selection for SLC, ABC transporters, phase 1 and phase 2 enzymes is very, very low. So the bulk of those genes here have low scores, indicating that they are not under major pressure and can even be homozygously for reduced functional alleles. Whereas on the other side, nuclear receptors are much more conserved. And the second additional fact that I would like to emphasize is that the vast majority of those variants that we identified here are indeed rare or very rare, with minor allele frequencies below 1% or even below 0.1%. So 96.2% of all variants, of all these 70,000 variants, had frequencies below 0.1%, and more than 50% only occurred on a single analyzed allele. So this really gives a flavor for the heterogeneity and the complexity of genetic variability in pharmacogenes. And the same is true also in drug targets. So again, close to 96% as reported in the other pharmacokinetic paper, pretty much the same numbers very large numbers of individuals carrying only or very large number of variants being only identified in one or very few individuals. And this here covers now 606 drug targets, which are all genes that are target of any medication that is currently approved by FDA. So now the question is, with this large complexity, how to actually make sense of that, and how can we interpret the functional impact that those variants are having? So how can we go beyond those common variants that are well understood and both functionally and epidemiologically characterized? And for the time being, computational algorithms are the only way to at least make a step towards this direction. However, while there are a large number of algorithms that aspire to predict the functional impact of genetic variants, pharmacogenetics has a couple of peculiarities, and I will try to convince you now that these common genetic algorithms are not accurate predictors specifically for pharmacogenetic variability. And the reason for that is that these algorithms were trained on disease-associated variants. So they were specifically designed to detect pathogenicity of a variant, but not the functional impact. For many genes, this is congruent, so almost identical, but for pharmacogenes, it is not. And the reason is that CYP2D6, for instance, serve, or many other cytochrome P450s as well, don't serve important endogenous functions. They might serve some endogenous functions, but no endogenous functions that would be directly associated with a given disease. And as a consequence of that, the positive training set on which those algorithms are tested do not include those functionally relevant variants in many cytochrome P450s and drug transporters, although they have functional impacts. And inversely, those algorithms use common variants as a negative set. The rationale behind that strategy being that if a variant is present on more than, let's say, 5 or 10% of alleles in the 1,000 Genomes Project, this is sufficient evidence that it's not causing a severe disease, genetic disease. However, again, for cytochrome P450s, there's a substantial number of variants that entirely abolishes function of the respective enzyme or transporter that do have frequencies above 10% or 5%. So again, indicating that the strategy to design an AI-based algorithm for the prediction of pathogenicity might perform much more poorly when used on genes with low evolutionary conservation. And this imposes major limitations. So with this in mind, we then set out to develop an algorithm that would be specifically developed and optimized for pharmacogenetic predictions. And for this, we used 337 variants with really gold standard experimental characterizations and quantitative experimental characterizations that were distributed in total across 44 different pharmacogenes. So this here shows one of those algorithms called Pruvian. In total, we tested 18. And you can see that those variants here, those 337 variants, some of them being experimentally neutral, some of them being experimentally deleterious. The gray box here indicates those that would be assessed by the algorithm or predicted by the algorithm to be functionally relevant. And as you can see, there's an overall slight enrichment of those deleterious variants in the gray box here. But you see a lot of deleterious variants that are not in the gray box, which means they would have been false negatives. And you see a lot of neutral variants in the gray box, and they would have been false positive assessments. So in the first step, what we did is we basically moved this threshold here around, shifted that up and down, and identified what is the optimal threshold now specifically for pharmacogenetic variants. And again, we've done that for all these 18 algorithms. And we see that for some, like polyphane and SIFT, it doesn't make much of a difference. So the threshold that was set already in the initial development was very similar to those pharmacogenetically optimized thresholds, whereas for others, such as West and GERB, there was a huge difference. And in the second step then, after this threshold optimization, we integrated all different constellations of algorithms into one Ensemble score using a random forest model and 5-fold cross validations. and assessed which is the best combination now using those optimized thresholds by integrating those different variants to predict specifically those 337 experimentally characterized variants. And the result of that, we showed that even after cross-validation, this ADME-optimized model indeed could improve the predictive power compared to the individual variants and individual algorithms and ensemble scores, and was specifically reliable, which is of course not very surprising, for loss of function variants and true neutral variants compared to those variants that only have marginal or slight effects on the functionality. And in addition, I would like to emphasize that the algorithm also manages to provide quantitative assessments. So not only can it predict whether something is neutral or deleterious, but it can actually make an estimate of the extent of functional interference. And lastly, as a disclaimer, I should of course add that this is still far from perfect. So overall predictive power is around 85 to 90%, but there's clearly further work needed to increase the predictive power, and specifically there's important need for clinical validation of those data. So as I mentioned here, this is experimental data that we use there. And I will come back in the last part of my talk to potential strategies of how they could be leveraged. So when we ran this connects specifically here to an assessment of star alleles, meaning those common and well-characterized functional algorithms. Again, it's reassuring to see that the ADME optimized prediction tool here, APF, shows the best predictive power. However, nevertheless, on those 365 variants with experimental annotations, overall there's still a lot of room for improvement. And so this is on pharmacokinetic genes. Now, if I go for pharmacodynamic genes, this requires a fundamentally different way of assessing variability. And here I would first like to introduce a very elegant study by the lab of Madame Babu from Cambridge and Alexander Hauser, who's now in Denmark. They analyzed the genetic variability in GPCR drug target, G protein coupled receptor drug targets. And they used a structural approach in which they then mapped the variability onto the crystal structures or predicted crystal structures, and identified that there was a large number of naturally occurring variants in functionally irrelevant sites, which could be binding to the arrestin or G protein, the intracellular domain, to post-translation modification sites, to the ligand binding sites, and so on. And this is what I find the most striking of their results. It's an exciting paper in general, but this is really indicating the power of this approach. Specifically, they zoomed in on the mu-opioid receptor, characterized 3 different variants, and compared it to the reference wild-type compound. Overall, they tested here four different compounds: the endogenous ligand, agonist, and an antagonist. So if you focus now on the red variant here, M153V, you can see that this is basically purely reducing the activity of any of those agonists as well as of the antagonist. So this is reduced function variant, if you wish. However, it gets more exciting when looking at the other 2 variants, the blue and the green one here. And specifically on the partial agonist and the antagonist, these variants, again, naturally occurring variants, are actually amplifying the response. And most surprising and striking is the response here to naloxone. Naloxone is a drug that is commonly used as in emergency care for individuals with opioid overdoses to immediately antagonize the opioid effect. And what you see here is that in those individuals that are carrying the variant, what those in vitro data would suggest is that naloxone, an antagonist, actually acts like a full agonist. So not only does it not help to relieve the opioid overdose in such a case, but it actually stimulates the receptor even further. So really nicely demonstrating that there are strong compound-dependent effects. And in a recent study, we extended that further to investigate all FDA-approved drug targets, also beyond GPCR receptors. and mapped the genetic variability and analyzed specifically those variants that were within 6 angstrom of drug binding pockets. And overall, that left 110 targets, 638 different drug pairs for which such high resolution crystal structure information was available. And so the first thing is what we see is that the variability inside drug binding pockets is lower than outside drug binding pockets. And when we aggregate the entire variability there, what we see is that the aggregated frequency of all those variants is a little bit higher than 16%. So that means that around 1 in 6 individuals carries one variant within a drug binding site, within 6 angstroms of one of those 638 drug binding pairs. And we then also went further to experimentally validate that. We chose ACE as one of the targets. And again, we see that the potency of those different ACE inhibitors across different variants differs by more than 1 order of magnitude due to the genetic variability in vitro assays. And this is corroborated by structural modeling approaches, then, which nicely confirm that the conformation and the orientation of those molecules is really substantially altered. Similar for BCHE, a cholinesterase. Again, we analyzed 7 different variants. 6 of those served as were entire loss of function. So we focus on the one that was not a loss of function in itself, but only slightly decreased enzymatic activity in itself. And then we added tacrin, which is a known substrate of BCHE, and drug used for the treatment of, or at least intended for the treatment of Alzheimer's disease. It's not on the market anymore. And what we saw is that this variant D98G indeed rendered those variant enzymes more resistant to tacrin mediated inhibition. And this variant is actually interesting because it was one of the very first variants that were reported as a pharmacogenetic predictor or biomarker in 1957 by Carlo and colleagues for succinylcholine-induced apnea, and has a frequency of 1.7% in the European population. So really indicating that this might be in the range of really having substantial population-based effects. And when we then collaborated with the Jan Korabecny's group in the Czech Republic, who are excellent chemists and very adept in designing structural analogs or derivatives of tacrin, specifically using squalene scaffolds, what we could see is that some of those could overcome this resistance, whereas others, for instance, this molecule here, actually inversed it even, and were now more efficacious and more potent in the variant enzyme compared to the reference compound. So again, corroborating on one side the compound's specificity of variant action, and on the other side, that this might also, such structural approaches might open the doors for a more personalized and informed drug development of analogous molecules. So now the next question I would like to address here is how to generate now training data with higher throughput, and that connects to the first half of my talk. And one very exciting strategy that emerged in recent years, really spearheaded by Douglas Fowler and colleagues in the US, is called deep mutational scanning. And in deep mutational scanning, one starts with a mutagenesis library that one can purely design in vitro approaches. Basically, it's a library that of plasmids that encode a given enzyme or transporter of interest, but is since it's a mutagenesis library, it's not a homogeneous, but it's actually a mix, so that each one of those plasmids encodes for a different variant. So one can cover the entire library of potential genetic variability in a given enzyme, meaning each amino acid being varied to each other possible amino acid in a single shot experiment. One transfects then in the first step this mutagenesis library into a cell model, sequences the initial diversity library, and then afterwards applies a selection assay. So for instance, when using a drug transporter, let's say ABCD1, also called MDR1, which transports many chemotherapeutic drugs, and then afterwards applies a chemotherapeutic drug to such an assay at the right concentration. What one can see is that those cells that are expressing a functional transporter can export the chemotherapeutic drug, and therefore viability of those will be increased. Whereas those that either do not express any transporter or express a functionally inactive transporter or a less active transporter will be either killed off, or at least proliferation of those will be reduced compared to those with a functional transporter. And therefore, one can then by sequencing the libraries after applying the selection assay and comparing those to the initial diversity library, one can check which plasmids now are and which variants, mutants, if you wish, have increased in relative abundance and which have decreased in relative abundance. And in the setup that I explained there in the beginning, using a chemotherapeutic in a multi-resistance transporter, one would expect to see that those that are encode a functional enzyme will be increased in abundance, whereas those that encode a non-functional transporter will decrease in abundance. And therefore, one can, in a single shot experiment, very elegantly evaluate quantitatively and experimentally, importantly, the functional consequence of thousands of variants in a single shot. This has been already successfully done for a couple of important pharmacogenes, CYP2C9, BQC1, TPMT, NOT15, CYP2C19, and Assoc1B1, and the number of these pharmacogenes is really increasing almost on a monthly basis at the moment. So one example that I would like to zoom in a little bit is CYP2C9. This was an assay that was then eventually conducted in yeast, and as you can see here in the bottom right, nonsense variants, so which means that introduce frame shifts or stop codons, they entirely abolish the activity, so which means they are entirely depleted, whereas synonymous variants, as expected, basically do not have functional effects. And missense variants cover the entire spectrum in between those, including increased activity variants. And this I find here is a very interesting and important validation experiment, where they then tested experimentally using gold standard assays, using two different substrates, the activity score that they evaluated in the high throughput assay, and compared those to the function and activity of the actual known candidate polymorphisms and haplotypes. And overall, I would argue that this correlation is indeed for both substrates reasonably good. And so the power of such a system is that with this massively increased number of variants, again, in the beginning we analyzed 337, and now with a single experiment one can generate data for 1,000 or even more variants on top of that. This provides a much larger database for training machine learning algorithms, be they random forest, deep neural networks, or others, and which therefore holds major promise for improving the prediction and predictive accuracy of pharmacogenetic variant predictions. And so in the last slides now, I think I still have 5 minutes. I would like to briefly give some indication of where we believe the field is currently and might be going to in the future, and what exciting opportunities lie ahead. So in the maybe that shouldn't be called past, but rather a different form of current. There's genotyping available that is in the range of tens to hundreds of variants that are properly functionally annotated variants that can already be used to make pharmacogenetic predictions. Whole exome sequencing is making major leaps and advances into the clinical implementation, and it's becoming more and more accessible with more and more available to both clinicians and pharmacogeneticists, and that drastically increases the number of variants that can be incorporated into pharmacogenetic modeling. However, the predictive accuracy of those becomes lower for two reasons. On the one side, that not all novel variants that are encountered can be made functional sense of, because the current computational pathogenicity prediction is not up to par. And on the other side, whole exome sequencing is missing important variants that are outside of exomic regions. And where we believe that the field might be eventually going, and this is specifically driven by technological advances in genome sequencing, is that also other genetic variants outside of coding regions can be included, and most importantly be made functional sense of. And that critically relies on the advances in both AI technology on one side and deep mutational scanning on the other side, to really provide ADME-specific functionality predictions that go beyond evolutionary conservation and also beyond the interpretation of pure missense variability, driven by powerful machine learning frameworks. So as conclusions, I would like to highlight that the human pharmacogenomic landscape is really heterogeneous and complex, including thousands or tens of thousands of naturally occurring genetic variants. Computational interpretation of pharmacogenetic variants really requires specific methods, both when comparing to pathogenicity associated genes, but also between pharmacokinetic and pharmacodynamic genes, because the modeling of gene-drug interactions and the modeling of altered enzyme function is in itself different. Variability in drug target genes is only in its infancy, so I think there are exciting studies ahead that could delve into further detail of those variants and their functional effects. It really remains to be shown what those exciting new AI-based structural prediction tools such as AlphaFold can put to the table and contribute towards increasing this particular pharmacodynamic predictions. Deep mutational scanning is a exciting technology to improve functional predictions, and I cannot stress enough that clinical trials are really of paramount importance to evaluate the benefits of NGS over comprehensive genotyping. And some trials, I should also add and emphasize, are ongoing to evaluate this, not on the NGS comprehensive scale, but at least across indications in a preemptive setting across Europe. And one that I would like to highlight is UPGX, called Ubiquitous Pharmacogenomics, whose results should be reported during 2022. And yeah, with this, I would like to thank everyone in the group in Karolinska. Also, since last year, I started an additional group in Stuttgart in Germany at the Institute of Clinical Pharmacology, and also thank the members there, and all funders, of course, and colleagues and collaborators, and you for your attention. Thank you very much for the invitation again. And if there are any questions, I would be very happy to take them.
Yes, Professor Laushke, I have a question. It was really an amazing presentation. And also I have seen that there is a huge knowledge gathered in terms of pharmacogenetics. While this knowledge is extremely useful for education and for clinical work, how do you see the commercialization of this knowledge? Because I think that commercialization of the knowledge in pharmacogenetics is a bit behind the knowledge itself.
Yeah, so for the commercialization, I can also refer to two different initiatives where I think there is major hope. That is on one side, the design and commercial dissemination of individual testing kits. And this can be both B2B, so business to business, or business to hospitals if you wish, but also business to consumer testing is really increasing in its prevalence, specifically in Europe and the US, but also China and other markets are really catching up. So which means that its empowerment of individual patients is really increasing. They're driven by commercial efforts. And on the other side, there are important efforts and endeavors to really bring this increased genetic decoding power, if you wish, driven by those platform developments, to incorporate that with interpretation based on CPIC guidelines, FDA guidelines, to make also that available to the customers and end users, both as on the level of physicians, but also at the level of individual patients. So there are major companies now that have extended their efforts into providing personalized genetic reports so that the patients can inform their physicians that, you know, I have this and this variant, and according to the best of pharmacogenetic knowledge at the time being, this puts me in a risk group for, let's say, carbamazepine.
So that was a very nice answer. What I see from your answer is that the patient centricity is important again, because the patient himself or herself should be knowledgeable about the pharmacogenetics first, what kind of pharmacogenetic features every patient has, and then go with this knowledge to the physician. If the physician is not aware of this and say, please, I have this mutation, or I'm a slow metabolizer of this, or I'm a fast metabolizer of this, and please be aware what you prescribe and how do you prescribe. Should I understand this in this way?
Yeah, so this is one side of the medal, but it's very important to have it as an integrative approach, in the sense that also the physician information is and education is of course important. So we know from the pharmacogenetic space that there is, or at least has been, some resistance that is associated, or at least was associated with a certain lack of training and knowledge of pharmacogenetics within the physicians, and particularly in primary care. So overcoming that, and to make the physicians really appreciate the added value that can be put on the table, this is essential. Otherwise, the actionability of those information, even if that's clear to the actual patient, might be limited. And again, physicians trust very much, and for all good reasons, clinical trial data. So, which again puts strong emphasis again on having unambiguous data there that demonstrates that there is added value that really can help when incorporating that information.
Thank you very much. This was a very nice answer. Thank you.
Thank you for the question.
Thank you. We also have one question from the audience here. It says, Volker, would you say that by using this NGS data, you might be able to find other pharmacogenomic targets for other diseases as well?
Yes, so if I understand the question right, this is about asking whether additional pharmacogenetic associations can be identified using NGS, and there the answer is clearly yes. So over the past years, there have been a couple of studies that have identified new associations for a variety of different drugs, be that for patient stratification in terms of toxicity, but also specifically on the response side. So in there are multiple examples for this, for instance, in immuno guided therapy. So yes, in short, absolutely, NGS opens up exciting new discovery possibilities. But as a side note here, I should also emphasize that this classical associations of, you know, one SNP with one phenotype, that might be more and more difficult to really translate or transpose to these new discoveries. And what I mean with that is that this is not feasible to analyze these singleton variants or ultra rare variants using conventional approaches that have been designed to assess associations of variants that occur with 10% frequencies, right? So one needs to have new statistical strategies, for instance, based on variant enrichment, and there are a couple of additional statistical frameworks to analyze that. So it's requires really advancements and adjustments at multiple fronts to identify such new variants and new associations.
Thank you so much. That was a wonderful discussion and presentation. Thank you for your time.